Namaste children. In the previous module, we had learnt about the monohybrid cross. Cross between two parents which differed in one trait, tall or dwarf. In this module, we'll be looking into a concept called as dihybrid cross. It could be defined as the cross between two plants with two pairs of contrasting traits. Let us look this concept with an example. Here, in this experiment, we are crossing two pea plants. One could produce the seed which is round in shape with green in color. This parent is crossed with another plant which is in wrinkled shape with yellow color. So we are crossing round green with wrinkled yellow. As per the Mendelian's dominance, the round and yellow are considered as the dominant characteristics. Could you just tell me the genotype now, children? Yes, round, capital R, capital R. I told green is not dominant, so it should be denoted with the alphabets small. So here, since the yellow is the dominant color, we are putting the small letter for this green. Small y, small y. When this is crossed with wrinkled, the shape of wrinkless or being wrinkled is not considered as dominant over here. So this could be denoted with small r, small r. Yellow is dominant color of a seed. Y, Y. When these two genotypes or the plants with these two genotypes are crossed, that will lead to an F1 generation which has capital R, small r, capital Y, small y. Okay, let's recapitulate the concept of monohybrid crossover here. There we had followed few steps, isn't it, children? The very first step was we need to identify the pure lines by means of continuous self-pollination. Whereas the second step was cross hybridization between true plants. The true plants or the true lines had happened. Here you obviously know these two are heterozygous parents. So now here the cross hybridization happened between these two plants. Let's recapitulate the steps involved in monohybrid cross, children. It involved the very first step of selecting the pure lines. Here, since we are going on with the heterozygous, we don't need to mind about the purity over here. Whereas the second step involved the cross hybridization. Third step, the self-pollination of F1 generation. Could you just track out which is an F1 generation over here? Yes, these are the parental generation. So this could be considered as the product of parental generation, otherwise called as F1 generation. What we're supposed to do now is we have to execute the self-pollination of F1 offsprings. This tick mark denotes the most dominant offsprings over here. Could you just find out which are the offsprings which fall on round yellow category, children? How many of them? Yes, 9. 9 of the offsprings are round yellow. Now could you just count how many just falls under this X? 1, 2, 3. The circle? 1, 2, 3. Triangle? 1. What is the conclusion over here, children? Capital R, capital R, capital Y codes for round yellow. Capital R, capital R, capital Y, small y also codes for round yellow. Capital R, small r, capital Y, small y codes for Capital R, small r, capital Y will also code for round yellow. Capital R, small r, capital Y, small y will also code for round yellow. What does all these things denote, children? Nothing. The presence of even one dominant gene will elicit the expression of only the dominant character. Accordingly, when you count for this X, what is very common about all these X marks, children? Round, small y denotes recessive color, hence green. Round, green. Round, this one, round yellow. Just to discriminate between round green and round yellow, I showed here. Round green. This round green. This also codes for round green. So you could see 3 cross which codes for round green. Dominant seed shape with recessive seed color. Circle. Small r, capital Y, small y. What is this, children? What could be the character or the phenotype out of it? Small r denotes the recessive shape. So it should be wrinkled. Capital Y denotes dominant color. Wrinkled yellow. This is wrinkled yellow. This is wrinkled yellow. This is also wrinkled yellow. So there are 3 circles which denotes wrinkled yellow. The combination of recessive shape with dominant color. You could see only one, the least product or the offspring which is recessive with both genes which are recessive. For example, wrinkled green. Wrinkled is considered as the recessive shape. The same way green is also considered as the recessive color. Can you understand, children? So the phenotype ratio of dihybrid cross, you should remember very important term over here is called as phenotype. This is not the genotypic calculation what we are crossing here, what we are doing here. So this is the phenotypic character. We are just counting down the character which is expressed. We are not at all counting about the genotypes which are present. Could you differentiate between the genotype what I am marking here and the phenotype which they express? Isn't it, children? We are not counting about the genotype. Rather, we are counting only the phenotypic offsprings, the offsprings which are produced out of the self-pollination of F1 generation. Now, the ratio is 9:3:3:1. Now, let me just ask you a question. What is the genotypic ratio of monohybrid cross, children? It is very good. 1:2:1. What was the phenotypic ratio out of monohybrid cross? 3:1. 3 were tall, whereas 1 was dwarf. Whereas here 9:3:3:1. You have to remember 9 denotes whom? Round yellow. Round seed with yellow color. Whereas the 1 denotes the recessive combination wrinkled seed with green color. Okay. Now let's see what is the inference of this dihybrid cross. Here I want to just recapitulate the inference which we got out of monohybrid cross. There were 2 laws of inheritance which were produced out of monohybrid cross. Isn't it, children? One was the law of dominance and the other was the law of segregation. According to the law of dominance, whichever is expressed in the F1 generation could be considered as dominant trait. According to the law of segregation, the gametes will receive only one allele of a gene, either tall or the dwarf. So here, this is the law of independent assortment. This is considered as the 3rd law of inheritance or the inference of dihybrid cross. So this is the 3rd law which comes after law of dominance and law of segregation, and this is the law which acts as an inference or which is the conclusion of dihybrid cross. According to law of independent assortment, in the cross between 2 homozygous, or you can even consider heterozygous individuals, because the combination or the crossing between the pure lines is not quite mandatory over here. For 2 pairs of contrasting characters, what we are considering? We are considering 2 pairs, round wrinkle, yellow green, of contrasting characters. We are considering seed shape and seed color. Okay, seed shape and seed colors are the contrasting characters which we are considering with two pairs. Two pairs are nothing but round and the wrinkled are the two forms of shapes. Green and yellow are the two forms of color. The inheritance of one character is independent of the other. Have you ever seen the assorted chocolates, children? You could see Cadbury, you could see the milk flavored, you could see the caramel, etc. Assorted. They are assorted or segregated independent of each other. The same way, you could see the parental phenomenon or the trait is not at all being maintained here. It is it is not only being maintained here. You could not see only the offsprings which are round which are round green and wrinkled yellow over here, which resembles the same as that of the parents. Rather, yeah. So, out of this cross, law of independent assortment is obeyed over here. Have you ever seen the assorted chocolates or the assorted sweets, children, where things are assorted? Different flavors have been assorted together. The same way here, you could see the genes have not been mixed up or they have not been blended. And you could also infer one important concept out of this is you can't see only the parental generation being inherited over here. So what were the parents which we chose, children? We chose two parents, isn't it? What were they? Round seeds which are green in color or crossed with wrinkled seeds which are yellow in color, isn't it? But in the offsprings of F2 generation, you could see all the combination of almost all the alleles, isn't it? You could see round seeds yellow in color, you could see round seeds green in color, you could see wrinkled seed yellow in color, whereas wrinkled seeds also in green color. This is the law which was proposed out of dihybrid cross, law of independent assortment. You should not miss out two pairs, contrasting traits, contrasting characters here. These two are considered as the mandatory keyword which you have to remember. Two pairs of contrasting characters. It's very important for law of independent assortments. We are employing these words because this is the inference which we got out of dihybrid cross. Now let's solve this activity, children. What do the progeny of a tall plant with round seeds and a short plant with wrinkled seeds look like? Okay, I want you to give only the genotype now, and I let you to do the Punnett square for this question. You have to find out the phenotypic ratio out of this dihybrid cross. Shall we just find out the parents, genotype of the parents over here, children? Yes. What we are doing, we are crossing tall plant with round seeds. So both the tall and the round are considered as the dominant characters, isn't it? So we can denote them with the capital letter. Capital T, round. Capital R. So you have to cross this with a short plant with wrinkled seeds. The short is recessive. Shall we put S over here? No, we should put the recessive form. So small t. So here wrinkled. Recessive shape of a seed. Small r. What will be the product out of this, children? Because we are cross hybridizing it, isn't it? So the product will be capital R, small t, capital T, small t, capital R, small r. What you're supposed to do now? Self-pollination of this F1 generation. So please do this activity and you have to find out whether this plant obeys the law of independent assortment. How do these traits get expressed? If I'm saying I'm so sad today, if I'm saying I'm so happy today, how you all come to know that? If I express it, isn't it? The same way, we all were talking about genes from the beginning. We were giving so many different terms for it. Sometimes as per Mendel, we call them as dominant, recessive, sometimes we call in general as factors, etc. But the thing is, how do we come to know these are the genes? We were talking about Gregor John Mendel in the last session. But even I used a word that he was so unfortunate since his work has not been accepted by most of the scientists, because he has not provided any physical evidence for the factor whichever he had identified. Factor is nothing but genes over here. Now, in this module, next to dihybrid cross, we'll be looking into how the genes are expressed, in what forms they are expressing their prevalence. Could you imagine what is this, children? These are the nuclear pores. We are entering into the nucleus. Now we are inside the nucleus. Hope you remember what is this shape which denotes. What is this, children? This is chromosome, isn't it? This is chromosome. These are the proteins which are present to protect the gene. Could you see? Deoxyribose nucleic acid here. Nothing but the DNA. A part of DNA is called as gene, which is converted into a protein. How could we define a gene? Gene is a part of a DNA which makes up the protein. In general, they make up a protein. Sometimes they do also make up the RNAs. They would even stop with the RNAs. Now let's see the process by which the expression of gene is happening. Genes are expressed in 2 steps. They usually employ 2 steps to express themselves. What could you see here, children? Could you see somebody is coming here and eating up the DNA? Isn't it? No. These are the enzymes. What are enzymes? What they are made up of? They are also the proteins. Proteins can function as a protein itself. Sometimes they could be enzymes. Some other time they could also be a hormone. So here, this is again a protein which is catalytic in function, which acts as a biocatalyst. You might have heard this word in your 9th standard. Now the thing is, this enzyme is present here to open up the DNA to produce the RNA out of it. Then the RNAs will be converted into the expressive forms of genes called as proteins. Let me clarify very important steps which are involved in gene expression over here. The very first step is gene has to be transcribed into RNA. Then the RNA has to be translated into protein. What you have to bother much about here is scription-lation. Transcription is something where the script is transferred from its higher version to its lower version, from complex English to simple English. Could be called as transcription. The same way, DNA, which is a nucleic acid, it is dissolved into its simplest form. It's called as RNA, isn't it? So the mechanism by which DNA is converted into RNA is called as transcription. The conversion of nucleic acid into a protein is called as translation, where the language itself is different. The nucleic acids are translated into proteins. Now, once the proteins are formed, they come out of the...
As each triplet is read, a transfer arc.
Once the proteins are formed, they come out of the nucleus and they will execute their functions. These proteins, whichever has formed out of this transcription and translation, could be a hormone or an enzyme, or it could be anything which may take up any functionality. Now, consider me as a gene. Now, consider me as a gene. Let's just look into the simplest form of how the genes get expressed. Consider me now as a gene, children. This tool as the protein. Okay, so consider this as a plant hormone. You all of course aware of what is the functionality of plant hormone, isn't it? It is involved in either the growth of the plant, sometimes it is involved in other purposes also. Let's consider this hormone is something which is involved in the growth of plant. That is, it is the one which decides the height of a plant. Now if I'm so functional, the gene is so functional, it produces this hormone. If this hormone takes up its action, then the plant will grow. If I am becoming so inefficient now, then I'll produce something which is also quite being inefficient, that when that is injected or when that comes into the play, obviously the plant growth will be retarded over there. Could you understand, children? This is the way when you could see or you could really feel how the genes are get expressed. Let's look on to a very important concept of how do genes get expressed. How do germ cells make a single set of genes? This is very important for you to understand the single set of gene. How do germ cells make a single set of genes from the normal 2 copies that all other cells in the body have? Let's have as an example, human as an example over here. Okay, let me just put a human cell. What is the chromosomal number in all of our body cells, children? It is 46. How many pairs do we have? 23. Excellent. Now what happens here is, even the germ cells or the cells here, germ cells are the cells which are present in the gonads, in the ovaries or in the testes, reproductive organs in simple. So the cells or the germ cells which were present in the gonads are called as germ cells. When these germ cells, they do have the same number of chromosomes as similar with all our body cells have. What happens now? What does the question asks you? How do they produce single set of genes? That actually means they are producing 23 plus 23. When these cells undergo a special kind of cell division, the gametes which were produced as a products will be maintained with only a single set of chromosome. So the special kind of cell division over here could be called as meiosis. You should not be confused with this term with mitosis. Okay, meiosis is the special kind of cell division employed by these cells to produce the gametes. So that results in the reducing the number of chromosomes. Here you could see 46, but the gametes are gifted with only half the number of chromosomes. Now let's look into a very interesting or fascinating process of reproductive cycle. How do the sexes are determined? Which decides the child or the offsprings to be either a male or a female? Let's take an example of paternal cell, which I am marking as P, and a maternal cell, which I am marking as M. This paternal, since it is from father, will have 2 sex chromosomes, otherwise called as allosomes. Isn't it, children? What are they? Very good. X and Y. Whereas here, X and X. What are the 2 types of gametes which could be produced out of these parents, children? Yes. X, Y. Whereas here, X, X. Shall we just dissolve the probability of offsprings which come out of this cross? It could be either X, X, or X, 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 Y, or XY. So the possibilities over here could be 2 daughters and 2 sons. Now the question is, who decides the gender of a child here? Either it is a father or it is a mother, children. Of course, the father is the one who decides the gender of the child, because he is the one who gives out the Y chromosome. That is the one which decides the offspring to be a male. Could you understand this process of sex determination, children? It's very important. Could you just tell me what kind of cell division this step involves? Of course, meiosis. Let's solve this question. Important question of the activity question of your NCERT. A man with blood group A marries a woman with blood group O and their daughter has blood group O. Is this information enough to tell you which of the traits, blood group A or O is dominant, why or why not? With this knowledge whichever you have acquired from the beginning of genetics, you would very easily say if the father has a blood group A and the mother with a blood group O, if the offspring, let me just put daughter, so I just put the capital letter D over here, is gifted with the blood group O, you would definitely say, ma'am, this the F1 generation daughter has just been inherited with the O blood group. In that sense, it must be an example for a dominant trait. But it's not. It's because the blood group does not obey the Mendelian laws of inheritance. It is an exceptional example where it falls under another concept called as multiple alleles. Could you understand, children? So, as of now, you have to remember blood group A is dominant. Blood group B is also dominant. Whereas blood group O is recessive. Blood group AB is codominant. So, blood group A is dominant allele. Blood group B is also dominant. Whereas blood group AB, some of you might be AB positive or AB negative, isn't it, children? That means both of the genes are expressing together. They are friends. They are expressing together. Co-dominance. Example for co-dominance. Whereas O group is called as recessive. Why it is called as recessive is because that denotes the phenotype which doesn't have any kind of sugar on its RBC. If suppose my blood group is A, that means my RBCs are decorated with the sugar A. If my blood group is B, my blood RBCs are decorated with the sugar B. If my blood group is AB, then that means I have both A and B sugar. And I'm an example for codominance. If my blood group is O, if suppose I'm proud to say I'm a universal donor, but my RBCs are not been decorated with any sugars. So in this lesson, to sum up, we have discussed with dihybrid cross and what is the phenotypic ratio of dihybrid cross, and we had even executed few cross. Then we had just come to a conclusion of how genes are expressed and what kind of cell division happens in the cells which are present in gonads. Then we had seen how the sexes are determined, and finally we had discussed a concept of multiple alleles where you don't need to go deep into the concept. As of now, you should be proud that A, B are the examples for the dominant blood groups, whereas AB is an example for codominance, whereas O is an example for recessive blood group. Namaste, children.